Okay, well, good afternoon and welcome back to Science in the Age of COVID-19. Today we are tremendously excited to have Florian Krammer from Mount Sinai. He is a professor of microbiology in the Icahn School of Medicine. Florian is the master of many things, among them antibody response to viruses, the ability to generate vaccines, T-cell and B-cell responses. Florian's Twitter account has been one of my primary sources of information on this virus. From that, I was able to watch as his lab flew into action beginning January 14th. They have been putting in long hours. It's a BSL-3 lab. They are working with live virus. So everyone has, stormtrooper outfits. It's really quite amazing. Florian's lab has produced many reagents that are going to be useful in the fight against COVID, as well as in many other diseases such as influenza separately. But what is probably going to be the most critical in this battle is his lab was either the first or certainly among the first to make, distribute, and fully validate a serology assay to be able to detect the robustness of antibody response against this infection. So Florian is going to give us an update on the antibodies that this infection produces and what benefit they provide. So Florian, you're up.
Thanks for this nice introduction. So yeah, I'm going to talk about antibody responses to SARS coronavirus spike protein. I disclaim I'm not a coronavirus virologist. I started to work on this in January. My lab is mainly focused on influenza, a little bit on arenaviruses and hantaviruses. So I thought I'm going to start with a little introduction. By now you're all SARS coronavirus 2 and COVID experts. Just wanted to start with where it all began. So basically in the end of December, the WHO, China notified the WHO that they had cases of pneumonia with unknown etiology in Wuhan, in the Hubei province. So Wuhan is south of Beijing and a little bit west of Shanghai. And initially, the idea was that these infections are associated with a seafood market that also sold wild animals in Wuhan. But basically, a few weeks later, we learned that there were already cases in the beginning of December, and a lot of these cases were actually not associated with the seafood market. So this virus is probably already for a little bit longer in the human population. It's going to be very interesting to see where it actually came from, and I'm hopeful that Chinese scientists will figure out when exactly it jumped and how it. But on January 10th, the genome of the new virus was released and uploaded on Eddie Holmes' blog. Fudan University was sequencing it, and then Eddie Holmes made it available. And that's when we started to work on this in terms of serology. Just a little bit of introduction. I don't have to tell you what a coronavirus is, but what I wanted to point out here is that coronaviruses are not really viruses that are new. We have 4 different coronaviruses that are circulating in the human population. This includes alpha coronaviruses, NL63 and 229E, and beta coronaviruses, including HKU1 and OC43. Those viruses are very likely all of zoonotic origin as well, just that they jumped long time ago into the human population. And then we have the bad guys, SARS coronavirus 1, which was causing an epidemic from 2002 to 2004, and MERS coronavirus, which is still jumping from camels into humans. And here on the right side, I have a model of the virus you have the genome on the inside, lost sense RNA that's covered by nucleoprotein. You have a lipid envelope on the outside, which is host cell derived. And in this envelope, we have an envelope protein, which is an ion channel, and the matrix protein. And then these huge, really humongous spike proteins. And this is why the virus is actually called coronavirus. If you look at this electron micrograph here, you see the spikes on the surface, and the virions look a little bit like a crown. And so the new coronavirus was sequenced and was placed on a phylogenetic tree very closely to the SARS coronavirus 1 from 2003. And that's why it was called SARS coronavirus 2. And the disease was called by WHO COVID-19. If you look at this phylogenetic tree here, MERS is quite far away. And then we have here the human beta coronaviruses, which are really far away. And then the human alpha coronaviruses, NO63 and 229E. So there's quite some genetic distance here. Just an update of today before I actually go into what we did in the lab. As of today, we have about 7.5 million official cases worldwide. There's probably many more unofficial cases, and about 420,000 deaths globally. If you follow the numbers, there's a lot of states in the US where cases are going up again. And then we have really a dramatic situation south of the border, including Mexico, basically all of Latin America. And we get increasing cases on the Indian subcontinent now as well. So just to go back to the virus itself, my lab, as I said, studies interactions between antibodies and glycoproteins. And so when the sequence came out, we decided to focus on the spike protein because, again, that's what I'm interested in. Glycoproteins are what I'm interested in for other viruses too. And here on the right side, you see a crystal structure of the spike protein. It's a homotrimer. One of the monomers is here shown in dark blue, and we have the receptor binding domain in red. So this is the part that actually binds to the receptor. As you have experienced, science was extremely fast with this virus and characterizing it. Already in January, we knew that this spike protein and the receptor binding domain bind to ACE2, which is the same receptor that is also used by SARS coronavirus 1, but also by NL63, which is a human coronavirus. And so we decided that this is going to be the target for assays that we're going to develop. Also because for other coronaviruses, we know that antibodies that target the spike protein are actually neutralizing. And we're interested in correlates of protection and functional antibody responses. And so we started to make these spike proteins recombinantly. You can see here on the upper in the upper part of the graph, a schematic of the wild type spike. You have a signal peptide, and then it's divided into two parts. The left side is the S1, which also includes the receptor binding domain. The right side is the S2, which has the fusion machinery. And then we have a transmembrane domain on the C terminus and an endodomain. And so when we made our constructs, we basically deleted the cleavage site between S1 and S2. So we replaced this polybasic cleavage site by a single alanine. And we introduced mutations that had been characterized by the Vaccine Research Center at NIH. And they had shown that these two mutations, introducing these two prolines, actually stabilizes the spike protein. And then on the C terminus, we just cut off the transmembrane domain and added a trimerization domain and the his tag so that we can purify this thing. And we also made a recombinant version of the receptor binding domain with a signal peptide and then a his tag on the C terminus. And so initially we expressed these proteins in different expression systems. My lab is using the insect cell expression system with baculoviruses quite a bit, but we also wanted to try the mammalian cell expression system. And so we expressed the spike, and you can see that here on the left side and the RBD in insect cells and in mammalian cells. And what we saw was there's quite a size difference, and that's because there's many N-linked glycosylation sites on the spike, and the insect cells attach much smaller glycans than the mammalian cells. We also have 3 glycosylation sites on the RBD, and that explains this size difference here. And then we started to set up an ELISA, which is, you know, a very simple and old-fashioned way to measure antibody responses to antigens, but that's what we usually do in the lab. And we had a pool of 50 to 60 negative control samples, which were pre-pandemic from late 2019. Some of them were from January 2020, and they were taken from 20 people who were 20 years of age to people who were older than 65 years of age. So we assumed that they would have experienced human coronavirus infections quite often. We also had control subjects in there that had influenza infections, chikungunya, hantavirus. So we had a whole collection of serum samples. And we were lucky, we had a serum sample from one person that had a diagnosed infection with NL63, and so we got serum from that person 30 days after the NL63 infection. And that is important because NL63, as I mentioned, binds actually to the same receptor as SARS-CoV-2. So we expected that if there's any cross-reactivity, it might come from that serum. And we also checked, and I will show you that, we tested some of these or all of these samples also for reactivity to human coronaviruses, human coronavirus spikes, and many had very strong reactivity. We also looked at IVIG preparations, and I will show you that data because IVIG preparations are made from donations from hundreds of or thousands of people, and the idea was that if you would see cross-reactivity that is highly prevalent in humans, we would see it there. We also looked as background into HIV positive samples from before 2010. Initially, we only had 4 positive controls. So we did this work already in February, and it was really hard to get samples from COVID-19 cases in the US. And so we got 1 sample from Finland from collaborators and 3 samples from Australia. And this really allowed us to validate the reactions and then start with the ELISA. Of course, later on, we had many cases here at Mount Sinai, and we included additional samples from severe cases, mild cases, and even from 1 asymptomatic person. And so what we saw initially, we did this old-fashioned ELISAs. You see the ELISA curves here on the left side against the insect cell-derived RBD and the insect cell-derived spike. And on the right side, we have the mammalian cell-derived RBD and spike. And you can see that there's more reactivity against the spike. There's more signal than against the RBD. For the RBD, we only tested the first 4 samples. We didn't go into the whole set. I will show you in a minute why. Again, there's much more reactivity against the spike than against the RBD. And then we quantified this initially, and what we see here is that we get background reactivity with the insect cell-derived protein, but we don't get background reactivity with the mammalian cell-derived protein. And the hypothesis is that there's some reactivity to the insect cell glycans, and that might drive it. And so we chose to move forward with mammalian cell-derived proteins. And specifically for the spike here, you can see that there's very nice separation between COVID-19 patients and the negative controls. As I said, we also looked at reactivity of our negative controls against spike protein from human coronaviruses, and I'm showing you here reactivity to 229E spike and the NL63 spike. And in fact, our negative controls had a lot of reactivity against those, but did not react with the SARS-CoV-2 spike. For the IVIG, we tested 27 different preparations, And again, those preparations are made from donations from many different, from thousands of different donors. And so if there is large scale cross-reactivity in the population, those samples would have shown that, but we didn't see any reactivity here. And you have to look at the lag.
Hey, Florian,
yes?
I hate to interrupt. Can you take a look at your mic and see if it's sitting a little funny? It's breaking in and out.
Oh, I'm sorry. I'm using my computer, so there's nothing to.
Okay, can you maybe pull it a little closer to you or something?
Yes. Is that better?
That's better.
Okay.
Okay, really sorry. Thanks.
Sorry about that. So basically, we didn't see any reactivity here with the IVIG preparations, suggesting that there's no cross-reactivity in the large-scale cross-reactivity in the population. And we also went to look at HIV samples. Again, these were 50 samples that were from 2010 or before. We tested that because there were reports that there's cross-reactivity from HIV positive individuals, maybe to these few peptides that look like they are derived from HIV in the spike, but that was really also not the case. And initially, there was a lot of confusion about which samples can be used if there's an influence of serum versus plasma, for example. So we looked at that as well, and we found that there is actually no drastic difference if you run these assays with serum or plasma. And we also looked at heat inactivated samples, and this was initially very important because a lot of people heat treat their serum samples to make them more safe. This is standard practice in a lot of labs when people work with human samples. And it was also reported that RNA of the new coronavirus was found in serum in some cases. And so initially there were reports that some assays didn't detect antibodies in heat inactivated serum samples, but would only work with non-inactivated serum samples. And we had that experience too. We tried a lateral flow assay that became available, and that didn't show any reactivity once we heat treated our samples. But you can see here that the ELISA is not really affected by that. And another point that we wanted, or another thing that we wanted to look at, and this was pretty interesting, is we wanted to look at the isotypes and subtypes of antibodies that react. So those are only data from reactive samples from COVID-19 seroconverters. And so what you see here is that we get a lot of IgG1 reactivity and a lot of IgG3 reactivity, but little IgG2 and IgG4 reactivity. And in a way that's expected. That's exactly what we see for influenza, for example, and what you see for a lot of antibody responses against viral glycoproteins. What was a little bit surprising is that everybody reacted with IgG3, but there's a fraction that didn't make IgG1. And we also have pretty good IgM and IgA reactivity. Now, what you need to know about IgG3 is that IgG3 is binding better to activating Fc gamma receptors than IgG1, but it also has a much shorter half-life than IgG1. And so that's something interesting. We are currently following up on that. And the other thing that I need to say here is you need to take these isotyping experiments with secondary antibodies with a grain of salt, because obviously the secondary antibodies are different. And so we are trying to confirm this now with other methods. And initially I said we chose the spike as target because we wanted to look at functional antibodies that could also neutralize. And so with a handful of samples initially, we already did a correlation analysis. We did microneutralization assays, and you can see the data here on the y-axis. This is basically neutralization activity, and then on the x-axis you see the ELISA endpoint titers against the spike, and we see a very nice correlation between those two, which was nice because that in a way allows us to measure ELISA titers, which can be done in a biosafety level 2 lab, and then extrapolate neutralizing titers. I'll get back to you in a few slides with more of that data. So this was all done in late February, beginning of March. And so we started to have cases in New York, and we were talking to our clinical lab, and they were very interested in setting up a serology assay for SARS-CoV-2. And so the initial reason for that was that there was no treatment, and groups in China had reported that convalescent plasma was something that could be used for treatment. But of course, if you want to treat somebody with convalescent plasma, you want to pick donors that have high reactivity to the virus. And so they wanted to set this up for this purpose. And so we transferred the assay to the clinical lab, to our colleagues there. And we got New York State emergency use authorization for using the assay, and also FDA emergency use authorization. The way we do it in the clinical lab is a little bit different than we do it typically in the research lab. So there's a first step where we screen at the 1:100 dilution for the receptor binding domain, for antibodies to the receptor binding domain. And the reason for this screening step initially was that we had very few patients that were actually positive. And we wanted to find the ones that had antibodies to the RBD, because we assumed that that would be the most neutralizing sera. And then those samples were then confirmed and titered on a second ELISA plate that had was coated with the full-length spike protein. And so that produces a titer and allows you to estimate how much antibody is there. And so initially, when this was assessed for the FDA, the clinical lab found 92% sensitivity and 100% specificity. I'll get back in a minute to the sensitivity, which we believe is higher. And the high specificity came about most likely because we have these two steps. So it's basically two assays that are done sequentially. And typically with any assay where you have that kind of setup, it enhances specificity. Just to talk a little bit about types of tests, there was a lot of confusion initially. There were a lot of serology tests that all of a sudden flooded the market. And a lot of them were binary assays, mostly lateral flow assays. And that's what I'm showing you here. There's basically two types of serology assays. You can have an assay with a binary outcome, so that's either positive or negative. And a lot of them are lateral flow assays. So that's like a pregnancy test, basically. You put a drop of plasma or blood or serum here, and then you have a control lane, and then you have a test lane. If the control lane shows up, that means that the assay worked. If the test lane shows up, you're positive. If it doesn't show up, you're negative. A lot of the assays, of these assays that came, that were on the market initially at low quality, low specificity, and low sensitivity. But there's other issues with these assays too, because if you want to assess if somebody is protected or not, or if you want to establish a correlate of protection, you might want to know how much antibody is there, and these tests cannot tell you that. But a quantitative test, for example, in ELISA, but there's other examples too, actually gives you a data, and that allows you to look at protection and at correlates of protection. And that was something that we wanted to do, and that's something that we're actually doing right now. I just wanted to point out that, so we wrote a protocol for a very detailed protocol for how to make these antigens and how to set up the tests in other labs. And we shared that first on my homepage, and then we also got it published in Current Protocols. It's open access, so everybody can get the protocol. And we shipped the reagents needed for setting up the ELISAs to about 250 labs worldwide directly from my lab. And they are also freely available from BI resources, and BI shipped a lot of these reagents as well. So what did we do at Mount Sinai? So this plasma donor screening really took off. By now we have screened 40,000 people by ELISA for reactivity to SARS-CoV-2. And these donors come, these are mostly mild cases, and they come in two categories. So we have people who had been PCR confirmed at some point. So they were sick, and then they were confirmed to have the virus. And then we have a second category of donors with suspected COVID-19. So they had disease that looked like COVID-19, but they were never confirmed by PCR. And so far, we have treated more than 350 patients with convalescent plasma. I'm going to show you data on the first 1,400 donors, and this is also from a manuscript that can be found at medRxiv right now. So as I said, there's two classes of donors that we looked at. The PCR confirmed donors, which we know had the virus. And when we look at them, and this is at any point in time, 99.5% of these people develop antibodies to the SARS coronavirus 2 spike. Only 0.5% in this case from this sample, it was 3 individuals, had not developed antibody against SARS coronavirus 2. Some of them take a little bit longer to make antibody, but eventually most of them really seroconvert. And the interesting part is the COVID-19 suspected donors. So these individuals had, as I said, disease, but they were never really confirmed to have the virus. And so in New York, the beginning of COVID-19 overlapped with the flu season. We had a pretty strong influenza B and H1N1 season here. And so what we see here is that only 40% of those people actually make antibody against SARS coronavirus 2, while 60% of them make no antibody. And we assume that they either had influenza or they had other respiratory infections that were not SARS coronavirus 2. So when people first come in for the screening, that's what this is. We get a range of titers, and that's what you can see here in A. There are some people who are negative at their first screening. We have very few individuals that have low titers, 1 to 80 or 1 to 160. And the vast majority have medium or moderate to high titers, 1 to 320 to 1 to 2880. And so these people who have lower titers or are negative are typically invited to come back 1 to 2 weeks later to get retested. And what we see there is that the vast majority of these people who get invited to get retested test positive when they come back for the second screening. And so the conclusion here is that really the vast majority of people that get infected with SARS coronavirus 2 make antibodies. There's one interesting finding that we had. So initially when people came for the screening, they were also tested again by PCR because we wanted to make sure that they're not infected anymore. And what you can see here is a graph that shows a number of tests on the y-axis and both symptom resolution on the x-axis. And we have 2 bars here, PCR negatives and PCR positives. And so what that means is that people who had recovered from the virus already for a month still in some cases were PCR positive for the virus genome. And I think this is what a lot of people see globally. We don't think that this is still a infectious virus here that we see. We think it's basically fragments of the genome, maybe dead cells and maybe immune cells that have engulfed virus. And there were just a few papers that came out, one from Erasmus Medical Center, I think on the 9th, that looked at that and actually didn't find any virus in people who were PCR positive after symptom resolution. This is just an overview of the testing at Mount Sinai. This is from the end of May. It's not up to date anymore, but the point that I wanted to make here was that we found out of 30,000 people, approximately 15,000 to be positive for antibody. And of these 15,000, 90% of them had a titer of 1 to 320 or higher. Again, indicating that most people make a relatively robust antibody response to SARS coronavirus 2. There's a minor fraction that doesn't respond well, but the majority really makes a robust antibody response. And so we started to talk to our clinical lab, and we started to get really interested in, again, the correlation between what we measure by the ELISA and the activity of these antibodies, the neutralizing activity. And so we now redid that correlation analysis that we had done in the beginning, but now with a larger number of serum samples. So we have 112 serum samples here that are spread across a titer range in the ELISA. As you can see, from negative to 1 to 80 and 1 to 160, which are low titers, and then 320, 960, and 1 to 880. And you can see that the higher the ELISA titer is, the higher the neutralizing activity of these serum samples is. Some of these samples have relatively high activity of IC50s of 1 to 1,000, 1 to 2,000. We even had some which are not in this graph that go up to 1 to 3,000, which is a relatively strong neutralizing activity. And on the right side, I'm showing you the percentage of samples in each of these titer ranges that had any neutralizing activity against the virus. And you can see that in the low titers, about 1 to 80, 1 to 160, about 50% of the individuals have neutralizing activity. If you go to 1, 320, this goes to 90%, and above that, everybody has neutralizing activity to various degrees, but every one of these serum samples had activity in the neutralization assay. Again, this is a neutralization assay with the authentic virus. This is not a pseudotyped particle entry assay. So the big question is, what does this all mean? And there were a few very nice publications in the non-human primate model recently, and I wanted to go into this. So these papers that came from Dan Baruch's group, really, really excellent papers that were published back-to-back in Science. And what you see here on the left side is basically a study where rhesus macaques were first infected with SARS coronavirus 2. Here you can see the RNA copy numbers in the lung. And you can see that there's a lot of genome copies initially, and then over time it goes down. And then I believe 3 weeks later, these animals were re-challenged. And what you can see is that there is, in the vast majority of animals, there is no replication in the lung anymore. And there's a blip here, which might be input virus. So the lung was completely protected from reinfection. The upper respiratory tract is a little bit of a different story, but for the lung, it's certainly the case. And the second paper that was published looked at vaccination with a homebrew and relatively ineffective DNA vaccine. And so what that did was there were different neutralizing a range of neutralizing titers induced, none of them really high, but was very heterogeneous. And what that allowed was to look at a correlation between the neutralizing activity in the serum and again the copy number in the lung. And what you can see here is a really nice negative correlation between copy number in the lung and neutralizing antibody titers. And that basically establishes neutralizing antibodies as correlate of protection in the non-human primate model. There was now an additional publication in Nature from George Gao's group that suggested if you give rhesus macaques a neutralizing antibody and then challenge, you can basically completely protect them from virus replication in the lung as well. And so this is in non-human primates, but there are a lot of correlates of protection in humans. And so I took this from a review by Stanley Plotkin, who is really one of the godfathers of vaccinology. And what you can see here is a list of vaccines and pathogens and correlates of protection. So we have bacterial pathogens here and viral pathogens. And for many of these viruses or bacteria, we know how much antibody you actually need to have in order to be protected. We know this for influenza, where we look at an HI titer of 1 to 40. We have this for measles, where we have a microneutralization titer of IC50 of 1 to 120. And then we have different ELISA units for hepatitis A and hepatitis B. And so the idea is now to actually look at that for SARS coronavirus 2. It would be very important to establish a correlate of protection for SARS coronavirus 2 for various reasons. And so to do that, we started a study here at Mount Sinai that's called BERRIS, which stands for Protection Associated with Rapid Immunity to SARS coronavirus 2. And so the aim of this study is to investigate if reinfection is possible, and that's still a big question. We all assume it's not possible, at least in the short term, but this is something that needs to be proven. And to also determine the protective titer. So if you get reinfection in somebody who has already antibodies, you can look at the breakthrough titers where reinfection is possible again. And so BERRIS follows individuals with and without antibodies to SARS coronavirus 2 for at least 1 year. Half of our participants are positive, the other half is negative. And so we collect serum from them every 2 weeks. And saliva is basically collected every week. And if somebody falls ill or doesn't feel well, they're asked to come in, and we check them also for SARS coronavirus 2 by PCR. And this has been initiated on April 27th. So for some of these subjects, we're actually already a couple of weeks into the study. And this is done with Viswanathan, who is a colleague of mine here at the department. There are initial longitudinal findings, not just from BERRIS, but in general from Mount Sinai. We see that the antibody responses are relatively stable. There's now people for which we have 3 months of data, and we don't really see a dramatic drop-off of antibody titers. That might come later, but at least for now, the antibody response is stable. And so far, we haven't seen any reinfections, and that's true for BERRIS, but that's also true for the Mount Sinai health system. Again, we had many patients with SARS coronavirus 2. The hospital is monitoring if any of those come back with SARS coronavirus 2 infections again, and so far that did not happen in thousands of people. Although initially there was a high attack rate in New York, which is now going down. And the last data set that I wanted to mention, and it's the first time I'm showing that, and this is just a teaser in a way, looking at seroprevalence. So what this graph shows here is basically the number of official cases in New York City. You can see in the week of March 8th, we start to see cases, and then the deaths follow basically 2 weeks later. And so we started to already collect samples from our clinical lab. So basically those are samples that are used for regular blood work and then would be thrown out. We started to collect them mid-February because we were very interested in seeing how many of these samples might actually have reactivity to SARS coronavirus 2. And so basically we have these sample sets now from 2 different groups. One group is in a way our positive control group, and that's emergency department, inpatients. And so those are basically samples from people who came in with acute SARS coronavirus 2 infections and then were hospitalized. And then we have a control or a background group or a screening cohort of people who came in for other reasons. There's a large group of OBGYN samples in there, cancer samples, people who had procedures and had taken blood before the procedure to match blood type to the blood donations, and so on and so forth. And so we started to screen those, and just to give you an idea about the number, for each and every of these weeks, we have about 150 in the positive cohort and we have about 400 to 500 samples in the screening cohort. And again, the positive cohort was a kind of a way of having a positive control in there, because we knew that this would go up, and it actually does. So if you look at the week of February 16th to basically March 15th, you see that there is a very low incidence of seropositivity, and then it really starts to go up relatively rapidly. And so March 22nd is a little bit of a delay of when we started to see an increase in cases in New York. And that also makes sense, because it might take some time until people seroconvert. And in this cohort, or in this group, the seroprevalence really shot up. And in the week of April 5th, we are at about 46, almost 50% in this group, which makes sense, because at this point, there were very few, non-COVID cases in the emergency department, and the inpatient units were full of COVID cases as well. If you look at the screening cohort, we see something similar initially. The percentages are lower, starting in the week of February 16th, and then we really don't see much of an increase until the week of maybe March 29th to April 5th. And in these groups, the seroprevalence only goes up to 10.1%. So we're still analyzing the data. We're actually continuing to taking these samples and analyzing these samples. The idea is to do it for a whole year. Of course, these two groups are pretty skewed. We would assume that this peak comes down again now, since we have many fewer cases. And this screening cohort here is a little bit more representative of the population, but it's of course also biased, because you would assume that, for example, pregnant women or cancer patients are extra careful with precautions against COVID-19. And so you would assume that the seroprevalence in that cohort is a little bit lower than in the actual population. And so in conclusion, we have developed a test to identify seroconverters. It's useful for finding donors for convalescent plasma therapy. There is a lot of analysis ongoing right now to see if plasma therapy was actually effective at Mount Sinai. The test is also useful for identifying healthcare workers that are already immune. We're not telling these people right now that they are immune against reinfection. I think that would be premature. But the truth is that if people learn that they're already positive and that they have high antibody titers, they feel pretty good about it. And it's kind of reassuring. It also basically confirms, if they didn't have a PCR confirmed infection, it confirms their suspicion that they had an infection. So it's in a way psychologically helpful. We can do serosurveys to determine true infection rates. And this is a good tool to understand dynamics of the antibody response to SARS coronavirus 2. We also found that the general population is completely naive to the spike protein of SARS-CoV-2 in terms of antibodies. I'm saying in terms of antibodies because we know that there might be some T-cell cross-reactivity, and we also don't know about the memory compartment of the B-cells because that's usually more diverse, and it could certainly be that there are rare memory B-cells that show cross-reactivity. They were maybe primed by infections with human coronaviruses, but have cross-reactivity to SARS-CoV-2. But memory B-cells don't make antibodies. The serum antibody doesn't necessarily come from that subset, and so that's not, that signal, even if there would be reactive cells, isn't showing up. This might explain the rapid spread of the virus, and it also questions if antibody-dependent enhancement could explain pathogenicity in 50-year-olds and older populations who are really at risk. That was a hypothesis initially, but we don't see enhanced reactivity in older people compared to younger people. Humans induce a solid antibody response to SARS coronavirus 2. This includes neutralizing antibodies and might lead to immunity. My conclusion here is this is not something special. It's not like Lassa virus where it takes a long time until you get neutralizing antibodies. It's also not a weird virus that wouldn't induce any immune responses. To me, it really looks like most other respiratory viruses that also induce relatively strong humoral immune responses. So in my personal opinion, SARS coronavirus 2 is nothing out of the ordinary. And there are important open questions, and I hope some of you are working on those too. One important question is, how durable are the antibody responses? I told you short term for 3 months, they're pretty stable, but we don't know what happens after 6 months or a year. We still don't know if antibody responses are protective against reinfection. Animal models suggest that this is the case, but it needs to be proven in humans. We also need to find out what titer is needed for protection. And as I said, there was already a very good paper from by Alex Sethi and Shane Crotty's group that looked at cellular immunity. But of course, there need to be larger studies to look into what role might cross-reactive cellular immunity actually play in protection of certain individuals. And so I'll leave it with that. I just wanted to thank my, my group here at Mount Sinai, and also my collaborators, specifically Viviana Simon, who is my partner in crime in a lot of these studies, Adolfo Garcia Sastre. We also have a lot of projects together, and then really a lot of people on the hospital side of Mount Sinai, Anja Vanberg, Carlos Cordon, Harm van Bakel, and Mir Sotillo, and also Katrina Kecierska, Jussi Hepojoki, and Oliva Palatti. So they provided samples initially in February that we needed to A, set up our ELISA, and B, actually validate proteins and reagents that we then shared and put into BEI resources. And if they wouldn't have shared their samples, we would have had a delay of at least 1 month with doing that. And so I think with that, I take questions.
Florian, that was amazing. We've got a lot of really good questions here. Sarada is going to start off with the Q&A.
Thank you, Florian. That was great. And let's get right into the questions. So first of all, do you have an idea of the time course of the appearance of different isotypes?
Yeah, that's a good question. So we didn't look in detail. There are reports that say that actually IgM and IgG come up at the same time. IgM peaks and basically goes down again. IgG keeps rising. We haven't looked in detail. The isotyping that I showed you was from samples, from very diverse samples. The earliest sample there was day 3 post onset of symptoms. And we already had at least IgG there, IgM and IgA. And the longest out was 30 days post onset of symptoms. But we didn't do kinetic studies yet. So that's something we are planning with a subset of samples that we already have here, but we haven't done that yet.
Mm-hmm. And while we are at the topic of isotypes, I mean, we're just wondering, what are the different functions of IgG1 versus IgG3? And is there any way to bias vaccine response to specific subclass?
Yeah, that's a good question. I actually would like to, so in mice, you can easily do that, right? You can switch from IgG1 to IgG2A to B, right? It depends on the adjuvant that you use, basically. In humans, that's not so easy. So IgG1 and IgG3 are similar in a way as they are both good at interacting with activating Fc gamma receptors, but IgG3 is much better at that than IgG1. Mm-hmm. And then there's the difference in terms of half-life, right? IgG1 is has a much longer half-life than IgG3. So those are basically the differences. And usually, I mean, IgG1 is called IgG1 because it has the highest prevalence in serum, right? In general, unspecifically, followed by IgG2, IgG3, and 4. And so that's kind of why I'm curious about looking at absolute titers or absolute concentration of SARS-CoV-2 reactive IgG1 versus IgG3. It's something that, as I said, we don't see for other viruses necessarily.
And since you mentioned vaccines, and since you mentioned Alexander Setty's study, so you have an excellent primer on vaccines, on the landscape of SARS-CoV-2 vaccines, and looks like most of the vaccine targets right now use a spike protein. But Setty's group recently showed that there's a good significant T-cell response against other epitopes as well, mainly the M and N protein. So do you think a composite mixture of epitopes would be a more potent vaccine target?
Yes and no. So I think the spike protein is an excellent target because it induces, as antigen, it induces neutralizing antibodies. And I think it's absolutely necessary. But yes, and I think I've criticized very often that every vaccine approach in the Western world focuses on spike and nothing else. Don't get me wrong, you also have CD8 and CD4 epitopes in spike, actually quite a few, right? But I'm a big fan of old-fashioned vaccines. So I think a live attenuated vaccine, for example, might give you the best of all scenarios where you have mucosal immunity and mucosal antibodies. And you might also get a pretty good cellular immune response, right? So I think more is better, but I don't think you want to have a vaccine that does not induce neutralizing antibodies. I think that would be bad.
Oh, and then here's another question from, I guess, Loren. Is there a correlation between antibody strength and seroconversion and other clinical manifestation, like, GI tract or?
Honestly, we didn't look at that. What I can say is that there is a somewhat loose correlation between disease severity and the magnitude of the antibody response. It's not absolute, right? So there is a preprint, for example, by Jesse Bloom's lab, where they looked at children which supposedly were asymptomatic, and one of those asymptomatic children, I think it wasn't asymptomatic. Anyways, it was not a kid that was in the hospital because of COVID. Had an amazingly high neutralization titer, right? So in general, if you have severe disease, we see a faster onset of antibodies and we see higher titers. But mild cases also make antibodies, and it's not basically black and white. There's some gradient there, and there's outliers.
And now we have a question from Kim Ritola, and I guess a lot of us have this question in our mind. What does the seroprevalence even mean? I mean, can we use, what role does antibody testing play in a safe return to work? Is there any reason to lean towards ELISA type of a assay, or can we get some idea from correlates of protection data? I mean, are there any other correlates of protection that we could also use along with the ELISA data?
Well, I mean, there's a lot of things that might correlate with protection, right? Some of them might actually confer protection. Some of them might just correlate with protection. But I mean, for flu, there's a lot of things that correlate with protection, right? And I antibodies, HI antibodies, neutralizing antibodies, CD8, CD4s. And so you could look at all of that also for SARS coronavirus too. It's easy to look at antibodies against the spike. It's much easier to do than, for example, to analyze the CD8 and the CD4 T cell response. In terms of what assays to use, I mean, if you do serosurveys, I think lateral flow assays that give you a yes or no response, these are point of care assays. You could even go to a household and test people there after needle stick or finger prick. So that's very convenient. If they're good, if they're highly specific and sensitive for certain studies, they're perfect, right? It's just that you don't get the level of antibody responses out of it. And that's what you need in a way to look at the correlate of protection.
Florian, on that note, I was doing some reading. Apparently you can detect IgG antibodies in saliva. And I mean, not always, but apparently people are trying to get HIV tests and some livestock diseases. They do serology on saliva. How possible do you think that is? Is that something that we should invest effort to try to get up?
So it's very possible, and it's one of the reasons why we actually collect saliva samples in berries. One is we test for presence of virus genome. The other one is we test for presence of antibodies in saliva. And that's super interesting because you can specifically look for antibodies, specific antibodies that have a secretory component. Mm-hmm. And the problem is a little bit that the titers are usually lower than in serum and they're more transient.
Yeah.
So again, I'm a flu person, right? And in the flu field, we're looking at mucosal immunity a lot. And that's actually something that everybody thinks it's the main driver of protection because that's where the virus lands, right? I mean, in the lower respiratory tract, it's more IgG1 that protects you. But if the infection starts, it's secretory IgA. But the problem is that the levels are often low. The signal to noise ratio in assays is also often low. You know, you start ELISAs or serological assays for saliva or nasal washes very often in a 1 to 2 dilution. Mm-hmm. But I think it's super useful, it's super interesting, but it's not as easy to study as antibodies in serum.
Yep, absolutely. You set me up well for the next question, which is, I think IgA doesn't really get enough attention. What do you think are the roles, good and bad, of an IgA response in disease outcome?
I'm actually not sure there's a bad role necessarily. So I think IgA, the secreted version of IgA is super important again to basically protect the upper respiratory tract. We don't know enough yet about SARS coronavirus 2, but a lot of these infections might start in the upper respiratory tract before they go to the lung. In other cases, there might be aerosol that's inhaled and goes directly to the lung. But if it starts in the upper respiratory tract, I think that might be the main factor that protects you, right? But again, it's not so easy. There's no direct correlation between basically monomeric IgA in serum and then dimeric secreted IgA1 in the respiratory tract. So I think there might be a significant role there, but we don't know that much about it. But that's also one of the reasons why, we talked before this presentation about monoclonal antibodies. And so where you want to have your monoclonal antibody in a prophylactic setting is probably on the mucosal surfaces, mm-hmm. and IgG basically doesn't go there, right? Or to a very low degree. And so that's why I think IgA is very important.
Yeah, what I meant by bad is mainly in the GI tract rather than the upper respiratory system. It's thought that IgA might be a primary driver of immune hyperactivation and maybe correlates with autoimmune responses. But so you said that this disease is a fairly traditional upper respiratory infection. I'm going to slightly disagree with that. There are a lot of reports of GI tract involvement. Certainly, I think the greatest mystery of this disease are the vascular phenotypes. There also seems to be rare but significant brain involvement with this infection. So, do you want to either agree, disagree, just take any sort of stab at that?
Yeah, I want to argue with you. So I think you're right. And most viruses that cause respiratory disease do other funky things too, right? What we see right now is you have a wave of infections. There's a lot of infections that go on at the same time. And that allows you to find things that maybe only happening in a small fraction of cases. Just because you have that wave, you have a lot of cases and you have a direct relationship. You can actually say, oh, you know, now we have an increase of cases of, I don't know, this or that. And I think that a lot of the human coronaviruses might do similar things, but because there's never a wave of cases, there's a trickle of cases, and usually there's very few severe cases, you might not actually find these things, right? As far as I know, there's also an association, a loose association between Kawasaki disease and some of the human coronaviruses. Now we see that also with SARS coronavirus 2, and it's the same. I think the reason why it's now, well, it might not be Kawasaki disease, but it looks like Kawasaki disease, right? So the reason why it's now easy to make that association is, again, because you had this huge wave of cases, and then this follows, right? If these cases wouldn't have come in a wave, but you would have, I don't know, 5,000 cases over the whole winter, you would have never found that association. So again, I want to argue that this virus is not really an extraordinary thing.
Okay, yeah, fair enough. We don't know. Okay, we got a lot of questions about potential genetic susceptibility that interplays directly with the measurements that you guys are making. You know, a lot of people have IgG1 or IgG3 deficiency. There's IgA deficiency. Do you, I mean, it seems like that is almost certainly involved with how people are progressing with the disease. Do you guys have any data on that, or?
No, unfortunately not. That's super interesting. But in terms of, we probably have the data, we don't have the analysis yet. So that's kind of a common problem. We're sitting on tons of data, but there needs to be time to actually sit down and look at different aspects. So I can't answer that question right now. It's super interesting. And I'm pretty sure there will be data that might or might not point in that direction, right?
Yeah.
I'd like to, here's one about neutralizing antibodies. And this comes to us from Abraham. It's actually interesting. He wants to know if there's any relation between symptom severity and neutralizing antibody titer. So I guess, yeah. Yeah,
so there is, as I earlier said, there is a kind of a relationship between disease severity and antibody titers in general. And we see very good correlation between what we measure as antibody titer and neutralization. So people who had more severe disease or who were sick longer have likely higher titers of neutralizing antibodies.
So along those lines, does the immune response from asymptomatic carriers, do you think, look any different from people with severe and severe to mild disease symptoms?
That's a very good question.
That also comes from Abraham.
It's hard to find asymptomatics, right? So initially we did find some just because, you know, they did contact tracing, and then you find people who are PCR positive, but they don't have symptoms, right? I think we had one initially, and that person made an antibody response. It wasn't the strongest antibody response, but it was a robust antibody response. But I don't have more data on that. It could certainly be that people who are only transiently infected for a short period of time make very little antibody. That's absolutely possible. But then it's also hard to prove. If you have a person who basically doesn't have antibodies, but also never tested positive, they could be an asymptomatic, but it could also be a non-infected person, right? So
hard to define.
Yep.
Oh, while we were just touching on the subtypes, here's a question. I don't know if we tangentially touched upon this, but Larry Cow wants to know if, did you observe any age-related difference in IgG titer?
Again, we have the data set, we didn't look at it yet. I think there was some data in an initial analysis that suggested that there's more antibody responses in older people, but and that might be biased by the disease severity, because the older you are, usually the more severe the infection is. But again, we didn't look really in detail. And again, as I said, Jesse also had some data that showed pretty high titers in kids.
Yeah, and on that note, Akiko has a really good paper out, I think, yesterday on gender differences of immune response. It would be really awesome to correlate that with your antibody measurements to see, gender differences in antibody response.
I mean, again, this might be biased, right? Because males suffer more from the virus than females.
Yes, it's going to be hard. Yeah, you would have to have a big data set and hopefully control for things like disease severity, etc. I think her paper is mostly on innate rather than adaptive immune responses, and there the differences are profound. So
I guess when you said suffer more, I guess you're referring to the disease severity, right? So males and
I mean, in general, men are always complaining more when they get flu.
That's a separate issue. We have another question that says, didn't the UCSF group, and I think they mean Joe DeRisi, maybe not, find a relatively large group, about 10% of people producing memory cells?
Wait, I wasn't aware of that. Memory B or memory T cells?
The question doesn't say. I don't know the reference, so.
I don't know about memory T cells. We didn't look into memory B cells necessarily. Of course, we did because we were sorting for isolating monoclonal antibodies. They're certainly there, but I don't know how long-lived they are.
Yeah. And from Larry, we have a question about so you've run the serology assay on 40,000 people. Are you getting a good estimate of your true false positive rate?
Well, it's probably pretty low. Again, the thing is that we coupled two assays sequentially, right? I can only tell you from the initial data sets that we had, where we looked at people who, just by the definition, because the virus wasn't around, couldn't have been infected. And again, we had 100% specificity there. It might not be 100% with these 40,000, but again, it's relatively hard to. It's easy to find false negatives if you have the PCR results from their when they were sick, but it's really hard to find false positives in a way, right, from that data set, because we just don't have that information. If somebody is positive, that person might have been infected, and there's no way to actually tell if the person was infected, other than serology.
Yep.
There's a question from Howard. It's a common question. Women make more interferon gamma than men do. So could that be influencing the antibody response, the difference in antibody response that you see?
Again, we haven't looked at differences between males and females yet. That would be something to follow up on, also in terms of the isotypes.
Mm-hmm. Do you have anything else, Loren? I guess I see pretty much most of the questions.
Yeah. Yeah, Florian, we'll just say again that that was spectacular. Thank you for taking your time out of fighting this disease to tell us about it.
Yeah, thanks for the invitation. It was a lot of fun.
Absolutely.
Yeah, likewise.
Thank you.
Thank you.
All right.
Bye.
Okay. Stay safe. Thank you again.